The other strange or peculiar member of the family is HER3. HER3 has been known to, yes, to bind ligands as other receptors, but it has a deficient kinase activity, a deficient kinase region. While the others have a very nice tyrosine kinase activity, ErbB3, since last year, it was assumed to have no kinase activity. But last year, there was a paper by the group of Mark Lemon in PNAS that commented that a little bit of activity, tyrosine kinase activity, is present in ErbB3. How ErbB receptors become activated? What we know about the mechanism of activation in which the extracellular region is involved and in which ligands play a role is the following. Ligands bind to what we call subdomain 3 first, and this provokes also interaction of ligands with subdomain 1. Ligand binding between subdomain 3 and 1 provokes a change in the structure of the extracellular domain, exposing this thing here that we call the dimerization arm, which is hidden in non-stimulated receptors. Remember that HER2 has already exposed this dimerization arm. This conformational change exposes the dimerization arm of one receptor that together with another receptor brings two ErbB receptors together. This is a type of receptor-receptor mediated dimers or dimerization. It is possible that there are higher orders of magnitude of more receptors being added to these complexes, but we will speak just about dimerization. This dimerization brings together the kinase regions, which are intercellular in two receptors, and which one kinase region called the donor kinase provokes activation of what is called the acceptor kinase. As you see here, this region here is quite symmetrical. This region here is not symmetrical. This is a mechanism of activation which is not symmetrical. The activation of the acceptor kinase provokes phosphorylation of tyrosine residues in the C-terminus, especially of those receptors, and this creates sites that act as docking sites for signaling molecules that will later on be phosphorylated or not, and will allow signal transduction by receptor activation. Why do we study ErbB receptors? And I already mentioned to you, well, we study ErbB receptors and their ligands, this system, because they are expressed in certain tumors, breast cancer, stomach cancer, ovarian cancer, head and neck, glioblastomas. Many different solid tumors have overexpression or otherwise altered activation of ErbB receptors. They are oncogenic. We know that from in vitro and some in vivo preclinical models. And because of this, because they have been found in many tumors, many human tumors, they are used, as you very well know, as therapeutic targets. What about expression? This is just a HERCEP test. This is a test that we use. This is a Salamanca sample of 2 breast cancer samples, 2 adenocarcinomas, poorly differentiated and a slightly better differentiated adenocarcinomas. There are pathologists in the audience, so please, if I say something wrong, please correct me, okay? When you stain with anti-HER2 antibodies, this upper tumor, there is very few staining. This is probably a negative tumor. Huh? What would we say about this? 1 plus or nothing? 0. No staining in the upper one. Okay? The lower one has this typical staining with the antibody that recognizes HER2, that recognizes the intracellular region of HER2, this typical cobblestone appearance indicative of membrane staining. Even if we amplify, if we magnify the image, we see the nice staining in the membrane, the nucleus, so we see a really nice picture of a membrane protein that is overexpressed in this patient. It is well known that overexpression of HER2 is linked to bad prognosis. I'm not going to stop here. This is an old slide, and you all know this. And this, the fact that we have overexpression of some of those receptors in human cancer led to the development of different strategies. And I put here a couple of them. I mean, there have been many different basic studies, for example, using monotoxin conjugates or doing antisense oligonucleotides, intracellular antibodies that can trap ErbB2 intercellularly to prevent it from coming to the cell surface, blah, blah, blah. Several different basic approaches, but only those two that I am showing here are the ones that have reached the oncology clinic, which are anti-receptor monoclonal antibodies and the small molecule kinase inhibitors. Some examples here and others coming. Pertuzumab, which is an antibody against ErbB2, is now moving through clinical trials. Interestingly, even though we know now a lot about the mechanisms of activation, I just wish to tell you that trastuzumab or Herceptin that is used in the clinic has been, is known now to interact with subdomain 4. I would never design an antibody to subdomain 4. I would rather design an antibody against the dimerization arm to prevent dimerization, but trastuzumab works very well, and there is no question that it should be used. I mean, I'm not questioning this at all. I'm just saying that if I would, based on the structure and what we know about how ErbB receptors activate, if I would need to raise an antibody, for sure I would raise an antibody to this region rather than to subdomain 4. And those anti-receptor antibodies and small molecule kinase inhibitors are being used in different pathologies in the clinic. In the case of anti-ErbB2 or HER2 reagents, in breast cancer, EGFR antibodies, head and neck, lung, colon, and pancreas, and more to come. Those are already approved tumors in which, of course, after proper selection of the breast tumors that are going to be sensitive, which is rather complicated, still complicated. Those are the tumors in which treatments against ErbB receptors have been developed. What about the ligands? There's been less work on ligands that activate the ErbB/HER receptors. What we know is that the EGF family of ligands includes all these members, about 40 in mammals, including all the splice versions of neuregulins, and most of them, except crypto, to my knowledge, and some GGFs, are membrane-bound factors that suffer proteolytic cleavage to be released. But you can find these membrane-anchored forms of growth factors in human tumors. In fact, and this has been done here by a colleague of mine, Juan Carlos Montero, together with Javier Hernandez, who is here. We tested expression of neuregulin, or one of these ligands, in one, well, this represents one breast cancer patient. In Salamanca, we did a large study, but here, just to show you this picture taken here at Valdebron, showing the expression of the membrane of one neuregulin, type 1 neuregulin, in one sample from a patient. The stromal cells are pretty much negative. We analyzed expression of neuregulins in a series of 124 patients in Salamanca, a total of 150 something samples, because we had some metastatic tissue from some of them, but in all 124 patients, and half of them expressed proneuregulin. If they express proneuregulin and they express also the receptors, it's possible that proneuregulin can activate in an autocrine or paracrine manner the receptors present in the cells that produce, or at least that are in physical contact with the cell that produces the ligand. But what is the clinical relevance of the expression of neuregulin? Well, the clinical relevance is the following. We analyzed from those 126, we had a nice follow-up of sufficient time of around 66 patients. Those that were negative for neuregulin had a better outcome than those that were positive for neuregulin. So patients, breast cancer patients expressing neuregulin had a worse prognosis than breast cancer patients that are negative for herregulin or neuregulin. And this very much led us into trying to get insights into which are the mechanisms what are the molecular bases that can explain why patients that have high neuregulin behave less well than those that have low neuregulin. And I can tell you that we have been doing quite a lot of work. I am going to present you today data about proteomic approach that we have followed to identify a new signaling intermediate in the HER pathway, in the HER signaling pathway. But we have been doing quite a lot also of genomic analysis, which I am not going to present today because they are not still finished, but we have quite a few signaling intermediates that we are now studying and that we have been that we have identified through genomic studies. I'm going to present you some data. I'm going to present you a story of how we identified a novel hair signaling intermediate using proteomic approaches. First of all, let me present you the model system that we use. The model system that we use is a breast cancer cell line. This is an estrogen receptor positive and HER2 negative, even though they have a small amount of HER2 receptors. HER2 positive, or not very positive, but the cells express HER2 and HER3, and they respond very well to neuregulin. If you have neuregulin, they proliferate nicely, very well. In 2002, we defined a new signaling route, which is the MEK5 ErbB pathway that is responsible for participating in the proliferative responses upon activation of ErbB2 receptors in breast cancer cells. And today, I wish to tell you the story of P180 and how we identify this novel signaling intermediate in the HER pathway. And we came to that while analyzing signaling along the studies that we were performing. We were very much interested because you know that the PI3 kinase pathway is being more and more involved in signaling by several receptors, including ErbB receptors. So we were basically analyzing AKT phosphorylation as a readout for activation of PI3 kinase. And since we are not a lot of people here, you can still record this. But I'm going to tell you how we got here. We got here because we were spending a lot of money in antiphospho-AKT antibodies from cell signaling, which is one vial is something like 450 euros and it's 100 microliters. And we were just doing so many experiments that we we decided that we could not keep spending so huge amounts of money, because it was like a couple of vials a month, and that's a lot for us. That's a lot. So we decided to generate our own antibody against AKT phosphorylated as setting 473, to use it as a readout of activation of ErbB receptors. And here's what we got with our antibody. So, with our antibody, with our anti-phospho-AKT antibody, we nicely saw that if we stimulate cells, MCF7 cells, with the ligand for ErbB receptors, we could identify nicely the phosphorylation of AKT in Western blot, but we also saw a protein of 180 kilodalton that was cross-reactive with the anti-phospho-AKT antibody. For a few months, we decided to ignore this. We were just exploiting the antibody to follow on phospho-AKT, and one day I said, well, why don't we try to analyze what is this? And we thought it was worth analyzing what was this, because the molecular weight was 180. 180 is ErbB receptors. It's the approximate molecular weight of ErbB receptors. So we thought, well, maybe neuregulin is stimulating phosphorylation of a serine residue in one of the ErbB receptors. And this was 3 years ago. And the first thing that we did by then, because bioinformatics was very hot and very fancy, was to check in databases whether any protein would be similar to the peptide that we use to prepare the anti-phospho-AKT antibody. And we detected this one, HBP2. HBP2 is a transcription factor which was cloned by Dr. Sergio Moreno, who, the world is very big, but Sergio Moreno was upstairs in our institute in Salamanca. So I went there and I told him, well, look, I have found a 180 protein, the peptide that we use to raise the antibody has quite a few residues that are similar to HBP2, so would you have the human homologue of HBP2 that you can give us so we can check? Well, to make a long story short, it was not HBP2. So the bioinformatics approach failed. We spent 6 months working on this, transfecting, tagging, putting in MCF7 cells. By the way, this is the expected molecular weight, and the actual molecular weight of HBP2 was 130. So that was the first problem that we had, and well, but even though when you have this degree of homology, you wish it must be HBP2. Well, it was not HBP. Second approach, ErbB receptors. Have to be ErbB receptors, 180 kilodalton, the perfect thing. So what we did was to, this is what we get in cell extracts, minus plus neuregulin, the same picture more or less, phosphorylation of AKT and the 180. If we immunoprecipitate ErbB2 or ErbB3 from MCF7 cells, we nicely see their phosphorylation upon neuregulin treatment, no signal of P180. So the second approach, which was one of the ones that I was favoring, failed. P180 is not ErbB receptors, no ErbB2, no ErbB3. And we confirmed that by doing cell fractionation experiments. ErbB receptors are membrane-bound in the full-length form. But, our protein, P180, when we stimulated with neuregulin, our protein was in the cytosol, was not in the microsome. So that meant that P180 was not here, was not ErbB receptors, was not a membrane protein, was a cytosolic protein. And when we reached this point, as P180 was found by a reagent that cross-reacted with AKT, etc., etc., we decided to look for things that were downstream of ErbB receptors, that were in the ErbB pathway, and that were 180 kilodalton. And again, Richter, mTOR, TSC. Well, mTOR is a little bit higher in molecular weight, but anyway, we tested, we spent, by the way, all the money that we did not spend in phospho-AKT, we started spending in other antibodies. That was a failure also. No one of them was P180. So the next thing that we did, that we did not want to get to that, because I never had purified proteins. I never wanted to purify proteins. But we had to start a purification strategy. And the first thing that you think is that, well, if you have a nice antibody that in cell extracts is able to recognize phospho-AKT and P180, and I'm bringing you these, please take note that this is kind of a doublet there. We will come back later to that. Huh? Diffuse band, a doublet. So, well, if you have this antibody, use it to do affinity purification in a single step. So we decided to immunoprecipitate. It immunoprecipitates nicely phospho-AKT. Actually, you get rid of other background bands. But no P180. It immunoprecipitates AKT, but no P180. So we failed that. But we then decided to move into a more complex purification scheme using and taking advantage of something that we had been learning through the process, which is P180 is a cytosolic protein. Well then, we will do a first purification step, separating microsomes and taking only the cytosolic fraction. And we took the cytosolic fraction and we passed this fraction in order to purify this protein. We passed it through a phosphoprotein purification column. We threw the microsomes and we took the cytosol. The cytosol was run on a gel, well, actually on several gels, but this one, even with the scratch that we had here, that the postdoc did on the gel, you can see that we got something here that was not present in the non-stimulated cells. Okay, we took this into our mass spec. Even our all equipment, 300,000 euros worth, this time gave us something. Gave us three proteins. Three peptides corresponding to three proteins. PREX, a ubiquitin conjugating enzyme, and JIP4. Available antibodies for number 2 and number 3. We purchased the antibodies. No success. No available antibodies for PREX. And I have to tell you that from the three proteins, actually, most of the peptides that came out from the mass spec corresponded, the red ones, the ones labeled with the red arrow, corresponded actually to PREX. But no antibodies to PREX. And that was a problem at that time. So, well, we decided for once to trust proteomics. Sorry for the people that works with proteomics. We decided to trust proteomics and go for this guy, for PREX. First thing that we did was to look into papers in the literature. Actually, at that time, there were something like 7 or 8 papers published about this protein, PREX. So, this is very easy. You go one afternoon to one quiet place with the 10 papers and you become an expert on PREX. And that's what we did in a couple of afternoons. We become experts of PREX in a couple of afternoons, but no antibodies. And even though we realized that there were two genes, PREX1 and PREX2, that coded for a protein that has a guanine nucleotide exchange factor activity, and there were only few isoforms published in papers. But if you go, or if you went to databases, you would see 3 different isoforms of PRX1, not published, only PRX1 isoform 1 was published. And there were 2 isoforms of PRX2 published and 3 present in the databases. But there was some hints that we could follow to start excluding some PRX isoforms, because by molecular weight, PRX1 isoform 2 was too short, as well as PRX2 isoform 3. We could not fully exclude this one by molecular weight, by expected molecular weight. The next thing that we did was to question in the MCF7 and other breast cancer cell lines whether we had expression by PCR of PRX1 and PRX2. And as you see down here, we only see, well, this is the APDH, PRX2, this is a neuroblastoma, if I don't remember wrongly, that we used as a control for PRX2. No PRX2 in the breast cancer cell line, only PRX1. So this is great because now we can delete PRX2. And after doing the PCRs and realizing that there was nobody in the world being interested in raising antibodies to PRX1, to keep going, we had to raise antibodies to PRX1. We decided to do antibodies to different regions that would first be able to identify more than one isoform, that would even identify, and we did this by homology, PRX1 or PRX2, even though PRX2 was not present, but could be useful for future studies, to have a pan-PRX antibody. And we raised antibodies to the terminus of PRX1 isoform 1 and PRX1 isoform 3, specific for each one, that we could use for doing Westerns. And this is what I'm showing you. This is just a characterization of the anti-pan-PRX antibody. By that time, there was a Japanese group that had raised an antibody that worked in Westerns. We were happy to get a few microliters from them. And well, what we got was an antibody to PRX in MCF7 cells. Well, that only tells us that we have an antibody to PRX, not that P180 is PRX, by the way. But we started to have tools to analyze PRX. The pan-PRX antibody precipitates better than the isoform 1 specific or the isoform 3 specific. If we expose enough, maybe you don't see here, there is some PRX isoform 3 also in MCF7 cells. And this is a demonstration that PRX is P180. We took extracts, minus plasma regulin, the same that we have seen until now. You see the doublet here. If we immunoprecipitate pan-PRX with the anti-pan-PRX antibody, we can see that the anti-phosphor AKT antibody recognizes PRX as well as in the cell extracts. So for us, this was a demonstration that, let's say, the demonstration was likely P180. Which is the role of PRX in neuroregulin signaling? Has it any role in MCF7 cells, which is the cell line on which we isolate, we describe PRX? When we performed knockdown of PRX1 in MCF7 cells, we got several clones with different levels. Clone number 2 is a clone that was in which we could not have knockdown, and it responds very well in terms of proliferation that we measure as MTT metabolization. The same happens if we count cells. This clone responds very well. If we knock down PRX, even clone 1 or clone 5, the response, the proliferation in response to neuroregulin is very much compromised. This indicates that it's likely that PRX is acting in the signaling pathway that drives proliferation upon stimulation of neuroregulin receptors. And what happens if we inject PREX1 knockdown cells in animals? What if we do a xenograft assay? This is what we get in one of such assays. This is the growth of tumors derived from MCF7 cells, wild type. Well, actually, this is not wild type. This is the clone number 2 that has, in which we have not been able to do the knockdown. The cells form tumors that are growing with time. If we do the same with the short herping for PREX clone 4, PREX clone 5, we see that the tumors do not grow or even decrease in size. We don't know why they should decrease. But in this case, that's what happens. This is a control of the Western of PREX in the different, in 2 different tumors from 2 different mice of the different subgroups. And this is what happens in human tumors. We decided to analyze PREX in a battery of samples from our tumor bank, and we saw expression of PREX, as you see, high expression in some of them, low or no expression, or no detectable expression of PREX in others. I need to tell you that we have not been able to analyze whether this is phosphorylated in these samples. This is what happens sometimes with samples from patients. You don't have enough material to determine whether it's phosphorylated. It's phosphorylated or not, and we try to do that. We tested whether those tumors with higher expression of PREX behaved differently, and what we got was the following. Patients with tumors bearing low PREX have a better prognosis than patients having higher levels of PREX. This indicates, and we did several statistical analysis in collaboration with our clinical oncologist, and actually this was a factor independent of other prognostic factors. So it was not linked to ErbB2 overexpression. It was not linked to estrogen receptor or progesterone receptor expression, even though we initially got it through stimulation of ErbB receptors. Okay. But what about phosphorylation of P180? Remember that we initially identified PREX because it was detected with antiphosphosetting, with the antiphosphosetting antibody that we raised against AKT. So we were so enthusiastic with the proteomics that we decided to go for the phosphorylation sites of PREX. Again, well, at this point, we had an advantage. We already had an antibody. We did not need to go through protein for phosphoprotein column. In a single step, we could immunoprecipitate PREX from cells stimulated with neuroregulin and run this on a gel. As you see here, this band, which is not PREX, and other bands around here are in the same position, but look, PREX, after stimulation with neuroregulin, we have run, this is a long gel that we have run a lot to separate, well, PREX from other potentially contaminating bands or bands that would be pulled down. Look that PREX here is a little bit displaced, and this is representative also of change in mobility due to neuroregulin. So we decided to take these 2 bands and pass again through the proteomics facility. We did 4 different experiments with different results. Along PREX, we got different phosphorylation sites being detected, and we only selected for further analysis those that in the 4 different experiments always hit it as phosphorylated, which we call phospho-M1. So these guys we got phosphorylation from them in the 4 different independent experiments. I insist that probably there are more phosphorylation sites, but we only, I mean, there were about 10, 11 that we detected, but we only concentrated in those 4 sites that consistently appeared through the different experiments that we call M1, M2, M3, and M4. Look that the M4 has this small sequence here, which is coincident with the phosphoate. I mean, if we compare this to HBP2, you will say, well, HBP2 is much better. There's a lot more of residues that are similar. But that one, M4, was the one with higher analogy to the antiphosphoic AT. At this point, we were so enthusiastic with antiphosphoic AT antibody that we decided to prepare antibodies to the phosphorylated forms of the 4 peptides. 4 antiphospholipid antibodies. And we got some surprises here. Let's see, if we stimulate with neuregulin, let's go to the M4 back once again. This is the very much similar to the phosphoic AT, the most similar to the phosphoic AT. In fact, the phospho-M4, if we stimulate with neuregulin, we detect phosphorylation of Tregs at this site. The same, which is located here, the same happens with the M3 site. Okay? However, the surprise was that if we stimulated with neuregulin while increasing M3 and M4 phosphorylation, it was decreasing M1 and M2. This is M2, this is M1. I can tell you that M2 is not a great antibody, or at least is not that phosphorylated Treg. I'm going to show you a couple of yields about M2, and please later on forget about it, because it's not a great site, or at least it's not greatly phosphorylated. But what is interesting is that we saw a contrast in phosphorylation and dephosphorylation, so we thought that neuregulin was stimulating a phosphorylation-dephosphorylation cycle of Tregs1. This is a time course of the phosphorylation, of the dephosphorylation of the M1 and M2 sites by neuregulin. Okay. Rapid dephosphorylation, that means activation, likely activation of a phosphatase. The phosphorylation recovers over time a little bit, and the same happens with the M2 site. This is the kinetics of dephosphorylation of M1 and M2. M3 and M4, M3 phosphorylation of 5 minutes, which is maintained, also decreases at later times. M4, nice phosphorylation at 5 minutes, which is sustained for quite a while. So 2 sites are dephosphorylated by ligand, and 2 sites are phosphorylated by the ligand. If we measure RAC activity, we have not speak a lot about this, and I don't want to get very much into this because we are not experts, but Tregs1 was an exchange, a GDP exchange factor that would activate RAC activity. And what we saw was that activation of RAC was paralleling the activation or the stimulation of the phosphorylation of the M3 and M4 sites, and was specular to the inhibition of phosphorylation of M1 and M2. We decided at that point to check whether phosphorylation at these sites. The idea behind this was the following. Probably the ligand is augmenting RAC activity. The net effect is augmenting RAC activity, and to augment RAC activity, the ligand needs to dephosphorylate M1 and M2 and stimulate phosphorylation of M3 and M4. To check this, in our model, we used MCF7 cells in which we infected different forms of Tregs1. This form, serine 313 alanine, is a mutant in which setting, the inhibitory setting, cannot be phospho or dephosphorylated. Site M4, which is setting 1169, was substituted by alanine also to inhibit phosphorylation by neuroregulin 3. In other words, what we did was to try to amplify or prevent activation of RAC activity by doing these transfections in MCF7 cells. One of the questions that we had to answer to the reviewers was, well, how, I mean, because we did transfect this in MCF7 cells in which we had previously knocked down PREX1. One of the questions of the reviewers was, well, did you have a short herping that would inhibit PREX1 expression in MCF7 cells? I mean, how this can happen? How can you re-express? I say, well, that's life. That's how it is. We do transfections and we again restore the expression of the mutant or the wild-type proteins that we tagged with me, okay? And as you see here, this is the response in terms of RAC activity of the wild-type minus plus neuroregulin, increase in activity. If we mutate, if we impede the phosphorylation at the M1 site, which is an inhibitory site, if we don't allow this REX protein under resting conditions to be phosphorylated, we see an increase in resting activity. This is indicating, we performed some statistics and this is significant. It's not great, but it's significant. So this is indicating that the M1 site is in fact acting as a negative regulator of REX activity. And if we mutate the M4 site, okay, we don't see a much decrease, but we compromise neuroregulin stimulated activation of RAC. That indicates, and that we take it to confirm, that the M4 site, phosphorylation at the M4 site activates REX. Phosphorylation in M1 site inhibits REX activity. So in conclusion, under resting conditions, M1 and M2 are phosphorylated. Now we know a little bit more about one of the kinases. We have been searching for this, and we now have got one kinase that activates M1, which is not PI3 kinase, or other kinases that you may imagine. We still don't know very well the mechanism of phosphorylation of M3 and M4. We think that PI3 kinase is involved, but we are not sure of this. We have a weird experiment. I can comment it here, just to see your feedbacks. We used, to inhibit PI3 kinase, we used an inhibitor that you are familiar with, which is BEZ235, which is a great inhibitor of AKT activation by neuroregulin. I mean, you add neuroregulin, and that beautiful band that shows up when you add neuroregulin, and which is AKT, you retreat cells with this PI3 kinase inhibitor, and neuroregulin does nothing, does not phosphorylate AKT. However, it still phosphorylates P180. And we do not understand. This is a quite clear experiment. Quite convinced, not that there are some doubts. Is that you blunt completely AKT phosphorylation and you still have almost intact phosphorylation of P1. So we are not very much convinced at this point that PI3 kinase is involved in M4 phosphorylation upon activation of neuroregulin receptors. So after activation, M1 and M2 are dephosphorylated, M3 and M4 are phosphorylated, and this drives proliferation, migration, and invasion. And finally, I wish to thank the people that is working in the lab, especially Juan Carlos Montero and Samuel Sebane, who did most of the work together with Alberto Caño, who is an oncologist now in the University Hospital of Albacete, and some other people that is working in the lab, Sicheng, Elena Diaz, Azucena Paris, who has now an independent research line, but in our laboratory, Stella, Marta Sanchez, Maria Jesus Ortiz Ruiz, who is now finishing and will read the thesis in a few days. And of course, I wish to thank the supporting institutions without whom I don't know for how long they are going to keep supporting us at this point, but I still have to thank them for the past, which is the Ministerio de Ciencia y Innovación, the Ministerio de Sanidad, through the, especially through the Spanish Cancer Centers Network program. And I wish to thank you all for your attention in this afternoon. Thank you very much.